title of my talk today is a residue-based correlation between equilibrium and rate constants is an experimental formulation of the consistency principle of protein folding.
So protein molecules fold into their unique compact folded structures, typically less than milliseconds, in a state of transition without passing through any metastable intermediate states. And importantly, this special property is acquired by evolution and optimized for each amino acid sequence. So in general, protein folding refers to the protein folding from completely denatured state U to natively folded state N. But our study can be applied to a more general case, two-state exchange of a polypeptide chain. So a variable part here, exchanging between states U and N, is embedded in a stationary constant region C, and the entire process can be described as a two-state exchange between states A and B. Protein folding study used to be a central research topic in protein science. So this plot shows you the trend in protein folding study. So it's a count. The vertical axis represents the count in PubMed database. So 15 years ago, so there was a peak here, and the surprise occurs in 1990s. So in those days, so a funnel-shaped energy landscape idea was presented, and this is a energy landscape here, and the vertical axis is free energy. So here, free energy gives energy, but after subtracting the conformational entropy S star. So here means the conformational entropy means because polypeptide chain has a large conformational variety, so it makes entropy large. And the horizontal axis or horizontal plane represents the conformational entropy S star itself. And from a particular state U, protein molecule falls on the surface, down on the surface, and the target is a natively folded structure N. And from different conformation U prime, so the trajectory does not cross to the other trajectories and fall down on the surface toward N. And this G star can be converted to the normal G value. It gives free energy of the system or a molecule. And the reaction coordinate is a horizontal axis. And this is a more familiar free energy level diagram. And the starting initial state is U, so it's a denatured state or less structured state. And the final state is N, a native state, and or more structured state. So because of this is a two-state exchange, so there's a high energy state here, so-called transition state. And importantly, this is also has a huge diversity of conformations. So we to emphasize this point, so we say ensemble, so TSE sometimes. So like this. And there are three free energy differences here. So this is activation energy for forward reaction. So this is activation energy for the backward reaction. And this represents the stability of the system. So free energy difference between the U state and N state. And the central topic of the protein folding was the characterization of the transition state. So the mutational phi analysis was embedded by Professor Alan First in MRC in England in 1992. So a mutation is introduced in the protein core to make a cavity, to create a cavity inside the protein core. So without changing any large confessor changes. And measure these three free energy differences for mutants and wild type. And phi is defined as this equation. So the numerator is a activation difference of activation energies from mutation to wild type. And the denominator is the stability difference here. And this delta means a mutant minus wild type. And this phi value is 0 if the mutated side chain here in state U in transition state ensemble, and phi equals to 1 if the mutated side chain in state N here in TSE. And the fractional value phi is obtained because this is the ensemble average over TSE. And if we make mutations to many amino acids, we can obtain this type of plot. And there are many data points here. And this slope represents the phi value. And if we make various residue positions, mutations at various residue positions, then we can obtain many plots like this. And to convert this one data to phi value as a function of the residue number, so we can identify folding nuclei here in the transition state ensemble. And this slide explains our research strategy. So to understand the physicochemical basis of protein folding, so proper selection of the coarse-grained level for observation is crucial. And the mutational analysis, phi analysis, uses molecular level, coarse-grained level for observation. But we selected a residual level, that is, amino acid residues as a local probe. And we found that dispersion, so say 5 or 6 years ago, the dispersion or distribution of residue-specific equilibrium constant K and residue-specific rate constant K of a bioactive peptide. And this is an example here. So nucleosin peptide has a 27 residue antibiotic peptide containing 3 monosulfide bonds. And after labeling with N15, so we recorded HSC spectrum, and this spectrum indicates existence of 2 states in solution. Then we discovered the correlation between the 2 residue-specific parameters. So this prompted us to start the protein folding study a few years ago. And this is an example here. So log K, small k versus log large K plot here. And this blue data is for forward reaction here. And this is orange data is for the backward reaction. So, and of course, due to the triadic relationship of 3 parameters, these 2 data are not independent. So regression lines cross on the y-axis here, and the slope here, the difference is equals to 1. And this plot is have another representation. It's a REFER, rate equilibrium free energy relationship plot. So because we can use this term and this term instead of these log parameters. So we discovered this type of residue-based LFER. So here LFER stands for linear free energy relationship. So here equation here, and this is a three parameters relationship. So there are three parameters and two equations. So this means only one single degree of freedom remains in this system. So this means the transition state must be closely related to the two ground states. So our question is, what is the physicochemical basis behind the correlation? And in 1983, so many years ago, Professor Nobuhiro Go, in those days he was an associate professor in Kyushu University, proposed the so-called consistency principle. And this is a physicochemical condition that makes the surface of the funnel-like free energy landscape perfectly smooth. So this can decrease the free energy of transition state like this to accelerate the folding or unfolding. And there are many different representations of the consistency principle, and this is one way is using a lattice Monte Carlo simulation. So lattice model is like this, and there are three rules here. So one, so no states other than states U or N exist on any folding path. That means no third structure or no native structures or interactions. And if native contact forms, so here we are thinking only a non-bonded contact interaction. If native contacts forms between two residues, so small attractive force here, but otherwise no interaction. So I want to explain using the 2D lattice model here. This is one of the state U here, and this is a folded native folded structure. And this black line represents the covalent bond, and these cyan colors are native contacts. And when we see the transition state here, for one structure here example, these cyan interaction, non-bonded interactions are seen in native contacts, in native structures. So this native contact is on, active to stabilize this structure. And in contrast, this orange contact is a non-native contact. This is off, inactive during this simulation. That means even if the atom 5 and 2s make contacts, but there's no interaction between them. So this model is so-called a native-centric model. So all the interactions are only for native contacts. So from any initial structures, so structure can be folded, is foldable to only one target native structures. It's a bit eccentric, maybe you think, but this is a model. A physical model should be very simple. And we have succeeded in elucidating, deriving some theoretical connection between the consistency principle and the residue-based LFER. And we published the paper in Biophysics and Physical Biology last December. And this journal is published by the Biophysical Society of Japan. And before I explain briefly the theory, I have to explain some remarks here. So we introduced a special subscript here, left arrow I and left arrow J, to represent the delta G of the entire molecule. So it's important. Entire molecule measured using a local probe. When we use this type of notation here, there's a risk of being confused with a local parameter. For example, phi I means it's a phi-value of residue I. It's a very local quantity. But truly, actually, this means so this value is of the entire molecule observed with residue I. And the second important point, so when we consider the system, we have to think considering the limiting case. So this is a simplified version of the energy diagram, and this is a typical one. So transition state must have the highest energy. When we move this transition state to the U state, it can approaches the transition state can approach to the state U infinitely here, and then finally can match state U like this. So this is so-called downhill falling. So very rapid falling without or transition state is here. But when in the reverse direction, we consider the transition state can approach to state N. So this is possible. In the limiting case, transition state still have the highest free energy, but N cannot match the state N exactly, because this is not the two-state exchange anymore. So we have when we define this rho NN value, I will explain later. So this parameter should be defined as a limiting case like this, using these special subscripts like this. Okay, so we have to think these limitations. And so this slide summarizes an ideal correlation we want to find in the LFER plots from the consistency principle. And so the consistency principle, by assuming the consistency principle plus the direction reversal of the reaction coordinate, so forward and backward reactions should be the same in a sense of the biophysical sense, of course. And there's so there's a chain direction reversal and the toriadic relationship between three parameters. So we can derive this, the basic equation we refer to as it's a rho ij. Rho ij is the slope that connects two data points in the LFER plot. Can be represented as it's an average of the two phi I and phi j residues. And this solution is a trivial solution is a LFER, it's a straight line, it's a linear FER. But we found the general solution is a parabolic curve like this. So we call this a new type of FER. So this is a quadratic FER or simply QFER. And to derive rho ij value, we assume this equation. So there's a four parameters here. These are constant value and are weighted by these phi terms here. And of course, phi I is the fraction of state N around residue I in the TSE, and the product is the fraction of the coexisting of N states around two residues in the TSE. And this is a weighted sum here. And so this is a forward rho ij, and this is a backward rho ij. They have to connect using this equation. And the toriadic relationship, we can obtain this form equation, and differentiating both sides with respect to log k gives this equation, equation 3. And substituting these two equations into equation 3 gives this equation 4. And this equation must hold for any values of phi I and phi j. So that is these coefficients must be 0 or just 1. So we obtain this equation, and I don't mention that rho UU can be obtained. Its value is 0, so because state U can match the state U, and this we can derive rho NN equals 1, and addition of these two parameters equals 1, and then we assume rho NU equals to rho UN. So because there's no reason they should be defined. So this one. So and substituting these values into the equation, this one, we can obtain the basic equation. Then we want to obtain a general solution of the basic equation, and we consider the limiting case again. So xi and xj, our two data points come close, very close, and we can derive a differential equation here. And by solving this differential equation, we can obtain this parabolic curve solution here. If A is 0, this becomes LFER, and the phi value is the slope of the tangent at the data point. And so, and if A is 0, then phi is constant, then of course LFER. And so in the left panel, so Newcastle data was analyzed by LFER here, two straight lines here, and this in this case phi is constant. So this represents the homogeneous TSE. When we analyze the data using QFER here, so we can obtain the variable phi as a function of residue number, and this represents a polarized TSE. And the phi value is a tangent of the slope, and then we obtain this real data here. And unfortunately, the measurement error was not so small, so large errors prevented us to discuss in details. But this slide shows you the phi-value analysis without amino acid mutations is possible. Now we discussion part. So at the mutation phi analysis is the molecular level. So even if we use tryptophan as a fluorescence probe, so in this case, of course, this is a local probe, but in general, this regarded as molecular level information. And this is a REFER plot. And in the mutation analysis, so this is a wild type and mutations. So that is different molecules. These are different molecules under a single condition. Or another type of molecular level LFER is a single molecule under different conditions. For example, different solvent conditions, water, alcohol, and so on. In contrast, our residual level observation should be used left arrow I here when using local multiprobes. So, for example, we use a backbone amide protein and observing by NMR, or indirectly hydrogen-deuterium exchange study can be used to monitor the backbone exposure of the backbone NH group. And by changing the local probes position, so we can see a variety of differences of these energy values, free energy values, and this the ratio of these two free energy differences is the rho value or phi value, we should say. And in this case, residue-based LFER or QFER. So in this picture graph, a plot, so different residues in a single polypeptide chain under a single condition. So this is a new type of LFER, so we call this residue-based LFER. And for ideal proteins under the consistency principle, so we obtain a good relationship, straight line here, but also real proteins. So the energy surface is minimally fluctuated. So this is a large surface. So folding rate decreases and some deviations from the perfect line. And we consider a non-foldable polypeptide chain, of course, very slow folding rate here and very large variations. Now I discuss the difference between the cooperativity and consistency. So cooperativity is inversely related to the dispersion or distribution of the log K values, residue-specific log K values. And in general, all log K have the same value, we think. And this is a full cooperativity, but in our opinion, it can't happen in reality. And the moderate dispersion of log K, so it means reduced cooperativity, and we think it is a realistic case. And a very large dispersion of log K, so this means no cooperativity. For example, in the case of non-foldable polypeptides such as ITP. And the consistency is the strength of the correlation. And the correlation coefficient R square is a indicator, but not a perfect indicator, but it's a good indicator for the smoothness of the structural changes. So please remember the smoothness of structure changes. So now I discuss the ideal proteins versus real proteins. So ideal protein follows the Go principle like this. So native-centric interactions. But real proteins are of course affected by many factors. So chain entropy in the loop and the tail regions, electrostatic interactions, and decelebration of hydrophobic residues. These real effects can deviate these data points from the ideal relationship. And this is a real example here. I want to comment. Not all the residues appeared in this plot. For example, the tip of residues of the N-terminal tail region here do not appear in this plot. This is because due to the small difference between the two chemical shifts between the two states, so XC peaks are unobservable and do not appear in this picture plot. And then there's an interesting, important feature here. So this nucleosine sequence can be segmented by different colors based on the structure. And here tail and hinge and the core region. And interestingly, these segments forms a cluster on the in the RF plots. So this means structure view and energy view are closely related. So according to the traditional view, one molecule can be described by a single parameters, and this perfectly cooperative. But our view is a bit different. So moderately cooperative, but highly consistent. So as shown in the previous slide, so there are some clusters structurally and energetically, and they may be called a foldon like this. These foldons have some communication to each other like this to be consistent. And if it's a single foldon, this plot forms a straight single relationship like this. But there are two consistent photons. They are apart in the plot, but they are on the same line like this. And of course, two independent domains, so they should be have their own different relationship. And I don't explain details, but there are some exceptional residues. So this is out of the cluster, but still on the line, so still consistent. And the another case is an outlier residue here. It's deviate from the consistent line here. So this means this outer residue has the non-native or third structure or interactions. But anyway, so the deviation from the linearity or QFER can provide the information of the structure and the energetics of transition states. And in the future, so because we showed the LFER and QFER for two-state exchange of a polypeptide chain like this, using XC NMR and the relaxation dispersion NMR and hydrogen exchange study. So, and also LFER can be found in the case of ITP binding to the target, and using the relaxation dispersion NMR method. So these examples are found in this paper. But we have not yet shown the LFER and QFER in the case of the typical protein folding case like this. So this is because no suitable method at the moment. The reason is this protein folding is a highly skewed equilibrium processes with very fast kinetics. So a critical challenge remains in demonstrating protein folding examples analyzed with residue-based LFER and QFER. And we suggest isotope-labeled residue-specific IR spectroscopy with the T-jump method, for example, or residue-specific fluorescence of non-natural amino acids cytodirectly incorporated into a polypeptide chain with a combination of the stop-flow analysis.
You are interested in our research, please read our papers listed here. So thank you very much for your attention today.